Now we can focus on different types of restriction and modification system or RM system. In different type of microbes, different type of restrictions and modification systems are present that can be categorized as type 1, type 2, type 3. So these are the main three types. And sometimes the fourth type, it is also called as type seconds. It can also be recognized in some microorganisms. Now, type 1 system was the first one to be elucidated or discovered from E. coli K12. तो जैसे पहले डिस्कस किया जा चुका है कि फ्रॉम द स्टैंड पॉइंट ऑफ जीन मैनिपुलेशन प्रोसीजर्स तो इसकी इतनी अहमियत नहीं है. Why? Because practically it is not good. The reason is that this type 1 restriction enzyme, it cleaves the DNA randomly. So if we analyze the structure of type 1 restriction enzyme, so mainly it is a multi-protein complex. So it composed of different type of subunits, and restriction subunit and methylation subunit, they belongs to the same enzyme. तो इसका मतलब ये हुआ कि एक ही एंजाइम है जो कि डीएनए को कट भी कर रहा है और डीएनए को मॉडिफाई भी कर रहा है. So basically it is composed of 5 subunits that included 2 restriction subunits, 2 modification subunits, and 1 recognition subunit. And for the proper functioning of type 1 restriction enzyme, it requires some cofactors like for modification, that includes the process of methylation of different bases, and as well as cutting, it requires ATP and the presence of S-adenosyl methionine. So it needs cofactors. As I told you that the same enzyme is responsible for cutting and methylation. If DNA it is unmodified, it is being fragmented, and once it has been modified by the same enzyme, then cutting is not possible. So this is one of the basic reason, and another reason that is they are not practically valuable is that they can recognize a particular DNA sequence but cut away from that recognition site and cleave the DNA randomly. So that's why they are not valuable in gene manipulation. The next enzyme, which is called as type 2 restriction enzymes, they are more valuable. The reason is that they recognize a particular target sequence. Now target sequence, it may composed of 4 base pair, 6 base pair, or 8 base pair. We are going to discuss after this topic. And within these recognition sites, these enzymes have the ability to cut the DNA into discrete fragments that can be used for further manipulation by using different type of vectors DNA. So another point is that this enzyme has separate modification and restriction activities. So separate enzyme for these two purposes, that is separate enzyme for restriction and separate enzyme for modification. Another benefit is that this enzyme don't require any cofactors to cut the DNA. So type 2 restriction enzymes nowadays, they are commonly used in the process of cutting and joining, especially cutting of DNA molecules. Then another type that is also not so much valuable, it is type 3 restriction enzyme. They are more closely related to type 1, although they can recognize a specific DNA sequence, and like the type 1, they cut away from the recognition site. So the DNA fragments that are generated, they are random. So desirable fragment length, sometimes it is not possible. So they are also not used in gene manipulation. The last category, which is called as type seconds, because they are structurally they are more closely related to type 2 restriction enzymes, but they are also have no value. इसकी क्या रीज़न है? इसकी भी रीज़न यही है कि although कि ये एक स्पेसिफिक डीएनए सीक्वेंस को या टारगेट साइट को तो रिकग्नाइज करते हैं, बट दे आल्सो कट अवे फ्रॉम द target sequence. So that's why they also have no value in the process of gene manipulation. So type 2 restriction endonucleases, they are mainly used. Why they are called as endonucleases? Because they can cut within inside of the DNA, not at the margin, within the central point of the DNA. So that's why they are called as endonucleases.